now that we've covered mitosis and also its consequence of cancer with uncontrolled cell division, we need to move on and look at meiosis. Now, mitosis and meiosis are very similar because they're both dividing cells. However, if you remember, mitosis just divides the body cells, so those somatic cells, and it's for growth and repair. However, meiosis is a special type of cell division that only occurs for sexual reproduction. And the terms are very similar, so mitosis and meiosis. And I should also mention here that I overpronounce these on purpose so that, especially since this is just an audio lecture, you guys know which one I'm referring to. If I were to say these correctly, it would be mitosis and meiosis, so they sound very, very similar if you pronounce them correctly, but I overpronounce them. Another way to keep them separate, especially on exams, as you're reading through the exams and you want to keep in mind if I'm asking about mitosis or meiosis. Meiosis deals with sexual reproduction. If you just look at the word sex and meiosis, they both have E as their second letter. And that hopefully will help you distinguish between mitosis and meiosis, especially on exams where it's written. So looking at meiosis and its purpose, it's just for sexual reproduction, and this is due to fertilization, since in sexually reproducing organisms, they give half of their DNA to the offspring and half is given by a partner. If both partners passed on all 23 pairs of chromosomes, like in humans, so it would start with 46 from each parent. Then each generation, the number of chromosomes would double. So 92, 148, 296, and then 592, and then it would be over 1,000 chromosomes in the next generation. And very quickly, you would have too much DNA. It wouldn't be able to fit inside the nucleus. So it's very important to keep the chromosome numbers the same. So 46 in every single generation. That's accomplished by starting with the 2 chromosomes from the female, 2 chromosomes from the male. Through the process of meiosis, we reduce the number of chromosomes to just 1 in the egg and just 1 in the sperm, or kind of 1 set. And then those are passed on to the offspring. So we have 23 pairs in the parents. Each of them has 46. Sperm and egg each have only 23. So during fertilization, it brings the offspring back up to 23 pairs, half from mom and half from dad, so that they have 46, and they match the number of chromosomes the parent has. And that's what meiosis helps organisms to do. Some other terminology we want to make sure that you guys know. Diploid refers to the number of chromosomes, and you guys are all diploid. So di means 2. Ploid refers to chromosomes, so this literally says 2 chromosomes. So diploidy would be 2 chromosomes. So you guys all have 2 copies of every single chromosome in all of your body cells. So every single cell in your body has a chromosome copy from mom and a chromosome copy from dad. Haploid means that, again, ploidy refers to the chromosomes, means that you only have 1 copy. The only human cells that only have 1 copy normally would be the gametes, or the sperm and egg, depending on which gender you are. Again, looking at some more terminology to make sure that we're all on the right track. Homologous chromosomes are similar chromosomes from the mother and father. So this figure here shows homologous chromosomes, and I will also draw it out. So we have homologous chromosomes. This blue one is from the mother, and we'll use red chromosome for the father. And you pick up that set of chromosomes, and they're very similar. So each set of chromosomes will have similar information on it. For example, maybe mom's chromosome at this particular location has information for eye color. Dad's chromosome, the matching one of this pair, would also have information for eye color, but maybe mom's chromosome has details that give you blue eyes, and Dad's chromosome has details that give you green eyes. The way eye color works, you'd be some combination of the two working together inside of you, but the point is that Mom and Dad's chromosomes don't match exactly. The only way that the chromosome from Mom and Dad would match exactly is if your parents were identical twins, and that's not possible anyway. So Mom and Dad's homologous chromosomes will be very, very similar, but have slight differences in the details. When these chromosomes start cell division, so we have a pair of homologous chromosomes in this cell, we start cell division for meiosis. Now, meiosis has two stages, meiosis 1 and meiosis 2. So going into meiosis 1, those chromosomes are going to make identical copies. And we've looked at those copies before. So Mom's chromosome over here is going to make an identical copy, and it will have, the same eye color information. Dad's chromosome is going to make an identical copy of his chromosome, again, with the same information as the original. So now these two matching chromosomes, the two from Mom and the two from Dad, those are sets of sisters. So sisters and sisters, or sister chromatids. And that's also shown in the figure here. These are still both homologous to this set. So these are homologous to these, and these are sisters, and these two are sisters. I may, depending on when I'm writing the exam, I may give you a blank version of this figure where it shows the two chromosomes, or maybe even just this part of the figure, and ask you to label the homologous ones and the sister chromatids. You would need to make it very clear that this set and this set are homologous, and then individually these two and these two would be sisters. So that's one of my common test questions that I would use. So you may want to remember that. So now that we have sister chromatids, those sisters in meiosis 1 are going to separate into two different cells. Notice the sisters are still connected, and then in meiosis 2, each of those sisters will move to an individual cell. So the reduction in reduction division actually happens here. So once we separate the homologous pairs, then we've reduced the total number of chromosomes in the cell. And we'll go through step by step everything we need for meiosis 1 and meiosis 2 to occur within cells. As always, we start with interphase. So during interphase prior to meiosis 1, our DNA is going to copy. So we're going to generate copies held together by centromeres of those identical sister chromatids. So here we have, see, 1, 2, 3, 4, 5, 6 sets of sisters inside this cell. And the blues would be homologous to each other, the greens would be homologous to each other, and the oranges would be homologous to each other. So we have pairs of homologous sisters in this cell. And like we did for mitosis, I'm going to continue using this same graph, and notice we're going through meiosis 1. When you're looking for meiosis on the exam or writing anything down, don't forget the numbers. You must number either meiosis 1 or meiosis 2. You must number it, otherwise you'll lose points for that. So as I go through meiosis, if it's exactly the same as what happened above, so we already know in interphase and meiosis we copy our DNA. If it's the same, I'll just leave this one blank. And pretty much everything that happened up here in mitosis all the way through will be the same, but we'll just add a few things. And I'll point that out as we go along. So we copy our DNA. So we've got our meiosis 1, we copy DNA. Again, just look up at mitosis. Since this is blank, it's whatever happens in mitosis. Moving into meiosis 1, again, it just refers to division of the nucleus. Meiosis is specific to that. So meiosis 1 starts with prophase, just like we saw for mitosis, but now we have to make sure it's numbered so it's clear if we're referring to meiosis 1, meiosis 2, or mitosis. And the additional thing that's going to occur during prophase 1 of meiosis is a process called crossover. So if you remember our homologous sets of mom's chromosomes, and we'll just shade them in blue, make sure they're the same size, and the homologous set of sisters that were inherited from dad. During prophase 1, what crossover means, these chromosomes would actually be kind of wrapped around each other fairly tightly, and during crossover, pieces of mom and dad's chromosomes get exchanged. So this and this will exchange pieces, and we'll have a chromosome that's partly mom's, and a piece of that's gotten cut off and moved over to dad, and then the chromosome that's still partly dad's, and a piece of it's gone over and attached to mom. So these two pieces at the ends have been exchanged, and that's crossover, and that always happens in prophase 1. So again, this is another of my favorite test questions, so I'll ask you maybe what event happens in prophase 1, and you would tell me crossover. Or I might ask you, when does crossover occur? And you'd need to tell me, well, it occurs in prophase 1. So just make sure that you're able to remember that these two occur at the same time. To add that to our chart, we have, we can see our chromosomes inside of our cell are crossing over. Our homologous sets of blue, orange, and green are all crossing over. And I need to point out right now, just to make sure, I only have crossover written in the box down here, but all of these are still happening as well. So the DNA is still condensing, the spindle fibers are still forming, and the nucleus is still breaking down. So all of these are still happening down here in meiosis 1, even though I've only highlighted kind of the additional process that occurs. Here's another figure that shows we have our purple and blue chromosomes. Crossover occurs, so they're laying close together, wrapped around each other. They exchange pieces of purple and blue, so now we have an entirely purple, and one that's purple with a little bit of blue, and this one is the opposite, so blue with a little bit of purple, and entirely blue. So make sure that you remember that that always happens in prophase 1. Moving on to metaphase. Again, we're in meiosis 1, so we need to label it metaphase 1. In metaphase 1, the main difference here between what we've learned already in mitosis is the chromosome pairs, so those homologous pairs, so the two blue ones, the two green ones in our diagram, and the two orange chromosomes are side by side. What we saw before when we did mitosis inside of our cell, the chromosomes were lined up right down the middle. Now these are sitting side by side with their homologous set of sisters. And that will be really important because that gives us independent assortment. And we'll look at that again on the next slide and also in kind of a recap at the end of this chapter. So because these chromosomes are sitting side by side, what happens is these spindle fibers coming in are only able to attach to one side of each of these. So the spindle fibers grow in and attach to one side of each. So when they pull and these move to opposite ends of the cell, the sisters stay together. And when they finish with meiosis 2, which we'll cover in a minute, they can have a variety of combinations, and that all depends on how these chromosomes line up. So you notice this blue is a lighter blue, here's the lighter green, and here's the lighter orange. If we assume that all the lighter colors came originally from the mother and all the darker ones came from the father, we can see that these are assorting independently. So the mother's original ones don't all line up on one side next to the father. So it's random which direction they get pulled to. So here is another diagram that shows the same thing. So we can maybe say the blue all came from the maternal side, purple came from the paternal or father side. They could, just by random chance, all line up together, and the offspring would end up with all blue, and these will be in separate cells. So these would be two separate offspring. Could have an offspring with all blue and all purple, or just by random chance, those first two could have switched. You get an offspring with one purple and two blue, or one blue and two purple. We can have the first two switch. So we have two purple and a blue, and two blue and a purple, or the middle one could have switched. So we have a blue, purple, and blue, or a purple, blue, and purple. So we can have all of these different combinations just with our original sets of 3 chromosomes. And that's called independent assortment, that the ones that came from the mother and the ones that came from the father in this particular organism will assort independently, go randomly to either side of the cell. And we will get a chance to look at this hands-on and work with it in class during the lab just before our exam on Tuesday. So if this is confusing, make sure you note that this is kind of confusing to you, and I'll make sure to go over it step by step with you and clear it all up before you take the exam. So again, in our chart over here, I've highlighted the differences. So instead of just the chromosomes lining up at the equator, which we saw before, now our homologous sets line up side by side at the equator. So we've changed this and just rephrased it, and now we've added independent assortment. So that's our metaphase 1. We have homologous sets line up side by side, and independent assortment occurs. When those spindle fibers start to pull those chromosomes apart, or the chromosomes walk along the spindle fibers, then that's anaphase 1, part of meiosis 1. And notice our sisters are still together. So we have all of our sisters' sets still connected, but we're separating homologs. So these two would have been homologous, these would have been homologous, and these would have been homologous to each other. We've separated those homologous sets, but the sisters are still connected, and that's really important as we move on through meiosis 2. So our anaphase 1 that we're going through, very similar to mitosis, except instead of separating identical sister chromatids, now we're separating homologous chromatid sets. So those sisters are still together. So sisters still connected as they move apart. Continuing meiosis 1 in telophase. Telophase, regardless of where it's happening in mitosis, meiosis 1, or meiosis 2, telophase is always exactly the same. It just reverses the primary steps of prophase. So we're going to reform the nucleus, break down the spindle fibers, and also the DNA is going to start to relax. And then it's followed by cytokinesis. I should mention here, depending on the type of cell, there are some cells that very quickly need to divide, and they may not totally complete telophase as they move into meiosis 2. It kind of depends on the type of cell, but for this class, just finish telophase and cytokinesis. Between meiosis 1 and down here, meiosis 2, there is a brief interphase. Depending again on the type of cell, some cells can be in this intermediate interphase for years, but there is an interphase. However, this interphase, no DNA replication occurs. So it does not copy its DNA again in this brief rest between meiosis 1 and meiosis 2. So here's our chart. Telophase, notice it's blank because it's exactly the same as what happened in mitosis. So nucleus reform, spindle fibers break down, and DNA unwinds, followed by cytokinesis, so the cytoplasm divides. So now we need to look at meiosis 2, and hopefully you guys already have a very good handle on mitosis, and I know I've just given you a lot of new information for meiosis 1, but if you understand mitosis, then meiosis II will be fairly straightforward because meiosis II is almost exactly the same as mitosis, with just a couple differences. DNA is not copied during interphase, and when we get down here to anaphase II, we're working with non-identical sisters. They're not identical anymore because of crossover. They've exchanged pieces, so now we're no longer exactly identical with those sister chromatids. And those are the only differences between mitosis and meiosis II. So meiosis II, again, very similar to mitosis. We've already separated our homologous sets, so we can see we're just coming out of meiosis I here, cytokinesis, so we've separated our dark and light, blue, green, and orange. And when they line up in anaphase, they're lining up single file, right down the middle. Only difference is this chromatid over here is solid blue, light blue. This one's light blue with a little bit of dark blue. So these two are no longer identical. Same thing with the green and the orange sisters in this cell, and also in this one down here. They're not identical to their sister anymore because of crossover. So when we move into anaphase, we're separating the sisters, just like we did for mitosis, but they're non-identical sisters. Finish up telophase and cytokinesis, and the end result is 4 unique haploid daughter cells. Haploid because they have half the number of chromosomes. They each have only one copy. So to finish off our chart, when we're looking at meiosis II, we can actually just ignore meiosis I because meiosis II and mitosis are so similar, except we don't copy the DNA. All this happens exactly the same. This happens exactly the same. Now we just have non-identical sisters because of crossover. This is exactly the same, and that's exactly the same. So mitosis and meiosis II are very, very similar.